certain important techniques which are very important in the healthcare. And basically the idea or the learning objective of this class is that whatever you have learned first 11 lectures, at least some of the concepts, if you can think how by learning the basics, you can solve some of the problems of healthcare. For example, COVID-19, many of you heard that COVID-19 infection is on the rise. So how will you identify the COVID-19 infection, for example, right? Or a person who is infected with COVID-19, how will you check the immune response? So you have learned the immune system, you have learned about the COVID-19. So how you will check the immune response against the COVID-19, right? You have learned about different cells. Can you differentiate between two different type of cells, or how will you do it, right? So those kind of questions we will answer. Besides, for example, you have learned about the DNA. What is the relevance of learning the DNA? Can DNA solve certain mystery of the biology? So that we will learn today. And final learning objective of the class is that what are the stem cells and how stem cells are important, what they do, where they live, what they are, and their relevance in the healthcare. So let us learn how knowing the DNA helps solving mysteries. I have shown you all the slides before, where I have said this Earl Washington in 1984, he was convicted for life for rape and murder, right? And after 16 years or 17 years, he in 2001, he found to be innocent with the DNA evidence. So what is in the DNA that was analyzed, and how it was analyzed, by which he was found innocent? Imagine the Earl Washington life. For 16, 17 years, a person is just jailed. And after the biology, the technological advances in biology, they have examined the DNA and found out as he is innocent. So what was analyzed? So the DNA, as we discussed, is basically certain sequences of ATGC which are aligned, right? And many sequences are there. One of them, what was analyzed was short tandem repeat, STR. So short means small, T tandem, you always say one thing is tandem means certain things are repeated one after the other. Repeats are repeat, of course. So certain sequence, a particular sequence, a unique sequence, if it is repeated multiple times, that is called STR. Okay. So how this forensic or genetic profiling is done? So genetic profiling or the DNA sequence profiling is generally done by obtaining the body fluid or a little bit of the tissue, because most of the majority of the body cells, except very few like RBC, they have the DNA, right? And those DNA can be isolated for body fluids or the tissues, and that genetic sequences or DNA sequences of that body fluid can be examined. And DNA testing that can identify individuals with a very high degree of certainty. All those sequences all are made by ATGC, but the way they are put or the way they are in the everyone's DNA is very unique. And these genetic profiles, they can be analyzed using short tandem repeats. And these short tandem repeats, which we will discuss what exactly they are, they serve as genetic markers. So what is these repeats and tandem repeats? So tandem, as I said, a group arranged one behind the other. And if you see this here, a random string of letters of the English alphabet is ALCD. But if you carefully just see in this particular sequence, what you will see but with the gem is repeated almost like multiple times. So if you just put gem and then this gem is repeated in this thing at multiple times. Okay. And another sequence, if you see where the gem is repeated, now tandem repeats. So gem. So these are called tandem because this 3 letters, it is repeated multiple times. Similarly, when you have the chromosome, and chromosome basically larger fragment of DNA which is coiled along with certain proteins, but if you stretch it, uncoil it, you will see these DNA sequences. And now imagine that this yellow particular fragment, which I divided in 9 different parts, if this one particular fragment has a sequence called TGCTG, these 5-letter sequences, and that sequence repeat now 9 times. Okay, so these 5 nucleotide sequence of ACGAC is repeated 9 times. Okay, and this is called short tandem repeats of certain nucleotide sequences in a DNA. So just for your information, if you have to clearly understand what is short tandem repeats, they are present in the human genome, our genome contains large number of STRs. They can serve as biomarkers because they are used in genetic profiling and to identify certain things. In this particular case, which we are learning, they are used for the forensic analysis. Each short tandem repeat is unique in multiple ways. One, nucleotide sequence. These STR can be somewhere between 2 to 5 bases. Length, how many nucleotides are there, right? Number of times that repeat is STR is repeated, like in the previous slide I have shown you 9 times repeat, somewhere it can be different. And the location where each STR is present, which chromosome and where exactly in the chromosome these STRs are present. Each such repeat may be present a certain number of times in an individual. And the number of repeats present also varies from person to person. So once you identify that in one particular person, these are 16, another maybe 19, or the location on the chromosome is different, that's how every individual you can identify with great certainty. That is the advantage. That's how these repeats are useful in the forensic science. The number of repeats, the 2 alleles of an individual also can vary, and these STRs are variations in the number of repeats of specific DNA sequences, and the probability that 2 people who are not identical twins, so in identical twins, yes, those can be similar, but if the twins are not identical, if they are, let us say, they are not monozygotic, they are dizygotic, or so they are, if not completely identical, they're such similar, then the probability of the 2 people having identical STR is exceptionally small. As of 2013, as the data what we have right now, that more than 300 innocent people have been released from prison as a result of this STR analysis of DNA evidence. So during criminal investigations, STRs at different loci at the chromosome, they are generally analyzed. So loci, you know that they are the position on a chromosome, right? And they analyze the DNA from the scene of the crime. That can be from the victim and the suspect, right? So somebody is a victim in a crime scene. You take the body fluid or the tissue from that crime scene person who can be the victim, and then whoever you suspect, you can also take the biological fluid or the tissue from that person. For example, here can be still be there. A small part of the hair one can get, and from the hair they can get the DNA, right? So take that DNA or the hair or tissue, isolate the DNA, and you can analyze. And in case of Earl Washington, they analyze the samples from the victim who made the charge, and then the Earl Washington and another person in the jail who was narrowed down as possible suspect. So now they can see if this possible suspect is the real criminal or the Earl Washington. So this is very important slide. So when they analyze the semen on victim, the DNA sequences were analyzed. Then they found that STR, the marker, let us think about marker 1, STR marker 2, marker 3, three different type of short tandem repeats they analyzed. There's position 17 and 19, STR to 13, 16, and 12 and 12. What they found that Earl Washington, those sequences were not matched at all. All three biomarkers, which are used STR1, 2, and 3, they all failed. But then Kenneth Ridsley, who was the possible suspect, identical match was found. And that clearly showed that Earl Washington was released and Kenneth Ridsley was then identified as the criminal. So basically, Earl Washington got relief because of this new advances, and because of biology, the basic biology they knew, and that's how the technology came in picture, that how technology can solve these problems. And now you can appreciate why imagine as an engineer, you can make a technology which can detect these three different biomarkers. But if you have no prior knowledge, what you were looking for, so basic biology as well as the basic understanding of the DNA and the understanding of the technology, then your thinking process then widens. And then as a good engineer, you can solve these daily or routine or the healthcare problems. And that's the reason you are taught the BB101 class. BB101 class is not just for mugging the biology. It's also appreciating or widen your little bit knowledge that how you can solve the problem of the biology or basic problem of the modern medicine or basic problem of medicine or healthcare, how you can see those problems and then use your engineering principle to solve those problems. Other basic concepts, for example, you have seen are the cell, right? You learn about the cell, you check its internal structure. So what are the different techniques which you can use to visualize the cell, right? We have discussed about microscopy. Many times we use the word TAM or SAM, or so different type of microscopy can be used. We also taught you, for example, I have mentioned when we were discussing the central dogma, and we discussed how the flow of information happens, right from the gene expression, the DNA to RNA to protein. And I was mentioning multiple times that proteins do everything, right, in the life. They are the functional unit of life. Because then why I am teaching you the protein, for example, right? Why you should know that protein are the functional unit of life, or why you should know protein at the first place. One of the reason now you can appreciate that in the COVID time, right? We are in the era of this COVID, and now you may be knowing somebody in your family or the distant friends or someone who are undergoing the COVID treatment or COVID problems, and you see their blood reports, right? What you see in the blood report, you see the cells, different type of cells, right? Somebody will call lymphocytes, somebody will say neutrophils, okay? And then what you see, another thing, you see the lung CT scan. So then lung images you are observing. Other things what you see, the proteins, the serum profile, so serum profile, antibodies. Now, another thing what you are seeing a lot, maybe thinking, or maybe you are hearing a lot about IL-6, interleukin-6. And they are saying if the interleukin-6 is high, then patients have higher chances to go under severity. And it will be very severe and person may die. Then you may be thinking, what is IL-6, right? And how it is measured, how you can quantify. All these blood test report, whatever you see, what are the techniques these guys are using to analyze these proteins, right? So this we will discuss very briefly. So do not worry that we are not going to give you a lot of information, but just very superficially. So at least in your normal life, if not for your deep understanding, but for normal life, you can appreciate what are the techniques used in the biology. So one of the technique which is very common in the diagnostic field, or in whatever the report you see many times, ELISA. And ELISA is something called enzyme-linked immune solvent assay. It's a plate-based assay technique designed for detecting and quantifying substances such as peptides, proteins, antibodies, and hormones. So anything related to protein, which is either the antibody or also type of protein, many hormones are type of protein, many peptides are smaller proteins. And interleukin 6, what, for example, is examined in the COVID patient, that also interleukin is cytokine, which is also a protein. So those are measured by the ELISA assay. So ELISA is used for antibody titers. Antibody titer is basically quantifying the antibody level. So if you are observing that, maybe you are listening in the television that Covaxin induces very high titer of the antibody. And now you are thinking, what is high titer of the antibody? And so antibody, now you know from the immunology class, that's a part of the immune system, right? But high titer means what? Level of the antibody in the serum. How those high titers are measured? They are measured by the ELISA assay. So ELISA is what? It's a method to quantify the proteins. So for example, if the COVID patient is there, they will have antibodies against COVID. If the HIV patient is there, in the body, there would be antibodies against HIV. If a patient is infected with Mycobacterium tuberculosis or bacteria, they would have antibody against bacteria. And how these antibody you can quantify? Using ELISA method. Okay. So detection of infection or immune response also against the COVID you can do by IL-6. So IL-6 is a cytokine, and this cytokine you can quantify by the ELISA. And these proteins are generally identified by ELISA, and this ELISA is multiple different types. Somewhere when the antigen is coated on the surface, then primary antibody, they recognize the antigen, and then primary antibody then are labeled with certain dyes which generally glow. And that mechanisms are again many different types. Sometimes it's chemical reaction, sometimes fluorescence, sometimes luminescence. So based on the where the primary antibody or where the antigen is captured or antigen is bound. So if antigen is bound and then primary antibody directly recognizing it, it's called direct ELISA. When the primary antibody is recognizing the antigen, but there is secondary antibody used to detect the primary antibody and be conjugated with the antigen, it is called indirect ELISA. When there is a capture antibody which recognize the one epitope of the antigen, and secondary antibody recognize different epitope of the antigen, and this mechanism when it's used is called sandwich ELISA, because the antigen is sandwiched between the two different antibodies which are recognizing it. And there's competitive ELISA as well. So there are different types of ELISA. This will not come in exam, but just for your information, because many times when you see these reports of the COVID, it is written the sandwich ELISA or the indirect ELISA or the competitive ELISA. So you should know that what is those kind of ELISA and how do they work. But in exam point of view, there will be no direct question to tell you what is indirect ELISA or sandwich ELISA. It's just for your information. Some of you may be thinking that when you have the blood, and in the blood, there are many different types of cells. In fact, not just in the blood, in our body, there are different types of cells. Some are small, some are big, right? Some look different, some have different protein expression profiles, some has different protein expression profile. So many of you are thinking that how these cells are identified, how you can identify that this is a, let us say, a monocyte, or this is a red blood cell, this is a T cell, this is a skin cell, another is an intestine cell. How will you identify this cell belong to which organ of this, what is the name of this cell and what are the characteristics of this cell? So there are different techniques to identify or visualize the cell. One is of course microscopy. Microscopy, you can either just visually see, and if by the visual inspection, you can identify the cell's internal structure or the cell surface or the shape, you may identify some cells. But many body cells will look the same. They will look alike. And then it is very hard to differentiate them just under microscope and without any special dye or special labeling, because they are similar, they're looking similar in the size, shape, and the maybe under the microscope. So you need different type of techniques to identify the cell. One of the techniques is called flow cytometry. What is flow cytometry? It analyzes a cell biophysical characteristics, as well as it also identifies the cell based on the expression of proteins on the cell surface. So it basically combines biophysical aspect of the cell, and biophysical aspect I mean cell size, mass, density, as well as the cell surface protein expression, which is biomarkers, right? So this technique in the facts of flow cytometry is very, very unique in quantifying and identifying the various different type of cells. And this technique right now is used for many different purposes. For example, HIV patient who ultimately develop AIDS. And why do they develop AIDS? Because they do not have any more the CD4 positive T cells. And because there are no more CD4 positive T cells, you can quantify them using this methodology. Okay, so if you see on the right hand side, the AIDS, this is a flow cytometry data. And what you see on the y-axis is CD4 cells, and on the x-axis the CD3. Okay, and this CD3, which are the P6 or P2 here, they are the CD4 positive T cell. In a normal individual, numbers are around 1000 to 1200 cells per microliter. In the AIDS patient, this number would be very, very low. And that's how you can quantify a cell, and then you can diagnose a disease. Similarly, cancer cell, for example, cancer, all of you know, this is originated from our own body. Our own normal cells become transformed at some point in life and they become cancer. So how you detect or how you differentiate between the normal cell and a transformed or a cancer cell or a malignant cell? So to differentiate between a normal cell and the transformed cell, you can examine their protein expression profile, which are proteins which are present on the cell surface. And the technique which you will use to quantify or to analyze or to examine these protein expression or cell surface expression of different receptors, one can use the flow cytometry technique. So flow cytometry basically it measures the single cells which are flowing through a detector system. So if you have the single cell suspension from the blood, you see in the top left there's a hand from the vein the blood is taken. So you have the blood. You take the blood in a tube, and in this tube you add certain antibodies which are labeled with a fluorophore, which are basically the fluorescently labeled antibody. And let us imagine you want to quantify the CD4 positive T cell. So you take an anti-CD4 antibody which are labeled with a fluorophore, and then you add in the cell suspension. And these antibody will go and sit on the top of the CD4 antigen. And now these labeled cells you pass through the flow cytometry machine, which has a laser. Basically, the single cell will flow in the tube or in the flow cell, and then the laser will throw the light, And because they are all fluorescently labeled, depending on the wavelength of the laser, this will absorb the light and emit at the different wavelength. And the emitted light will be detected by this optical system, which is the detectors. And this then converted into the, this signal will be converted into the statistical type of mathematical signal, where we can analyze the data on our computers. So basically, flow cytometry is a very powerful technique for characterizing the cells. They can be the immune cells, they can be the any body cells which you want to characterize to understand their surface phenotype. You want to know which proteins express on their surface, even if you want to know whether the COVID-19 related some biomarkers are expressing, or the AIDS or HIV related biomarkers are expressing or not. You can use the flow cytometry. So overall, flow cytometry allows the detection of certain surface biomarker on the cell. Intracellular factors is a beautiful technique to even see the inside the cell, what kind of proteins are present, how much they are present, and how many. So flow cytometry is a wonderful technique to detect the protein, to check the DNA content inside the cells, on the surface of the cells, and more importantly, in a qualitative as well as in a quantitative manner. So now let us talk about the stem cells and why they are important and when they are important. So basically, stem cells are used when there is a person born with certain genetic defect, and using the stem cell, you can regenerate that organ which is damaged, and for the regeneration purposes, you can actually get rid of the genetic defects in the humans. Here is a very touching story of a blood brothers for life, which is a family story. Okay. Let me read it for you. When Julie and Jonathan Henderson, they found out that their two-year-old son, Nicholas, had T-cell lymphoma, they were devastated. So they had a little boy, 2-year-old boy had lymphoma. Lymphoma is a type of cancer. They had just found out that Julie was pregnant, and what was supposed to be an exciting time preparing for the new baby that turned out into months of doctor visit, hospital stays, and chemotherapy. Right? After Nicholas' chemotherapy failed to work, Henderson doctor tried a relatively new transplant procedure using stem cells taken from the umbilical cord of the blood of their newborn baby, Nathan. Okay? So basically, the mother was expecting Nathan, and that's the same time Nicholas was diagnosed with the cancer. And within 2 years, when Nathan was born, then the cells from the umbilical cord of the mother, they were kept, they were used for the Nathan's treatment. So they worked with CryoCell International, which is a private cord blood bank and fastest growing umbilical cord blood bank stem cell banking company. They process their stem cells, test them, and store the Nathan's umbilical cord blood, and use this blood of the newborn baby to treat the cancer of the previous child. And now Nicholas is happy, energetic 4-year-old who is in the remission. Remission means cancer-free. He and his baby brother Nathan, they share a special bond. So basically, now imagine the stem cells from the newborn that saved the life of the previous sibling. So what are these stem cells? How they fix the genetic effect? But stem cells have a lot of regenerative capacity, and they can fix those genetically defective cells. You can get rid of those genetically defective cells, use stem cells to regenerate that particular body part. In this particular case, blood. So by definition, stem cells are very simple, right? They're able to divide in an asymmetric manner as well as symmetric manner. They are capable of differentiation. Differentiation here means that they give rise to different type of cells. So for example, if the stem cells of the blood, the hematopoietic stem cells, HSC, that will give rise to multiple different type of blood cells. Okay? Their daughter cells can remain in undifferentiated state or differentiate. Here you see that the stem cells you see in the right, it is dividing, and then the another daughter cell, it can have the self-renewal capacity, or it can undergo the differentiation. And so, for example, epidermal stem cells means which are present in the epidermis. Epidermis is the outer layer of the skin. So skin, you all know that skin continuously regenerate. So it needs a lot of stem cells, right? And hematopoietic stem cell, your blood continuously needs the new blood supply, new cells in the blood. That's why hematopoietic stem cells are there in the bone marrow. Various different type of stem cells based on where they are present and what stage of the development they are present. So totipotent cells are right coming from the fertilized egg, or they're from the embryo that are less than 3 days old. They can make the total human being, placenta and all other tissues. So they're highly regenerative and highly differentiating power in them. Pluripotent cells, once the embryo crosses 3 day old, from 5 day onwards, these are the cells which are pluripotent. They can make many things, but they cannot make the placenta. So embryo and fetuses that are older than 14 days, they do not contain pluripotent cells, and these cells can form every cell type in the body, but not the placenta. However, totipotent, they can even make the placenta. And then there are finally the multipotent cells. And this is very important for you to know, because the other two types, you require the embryo. But for multipotent stem cells, they are adult stem cells. They are present in all of us. As I said, for example, in the skin also has the stem cells, adult skin, I mean. So for all of you and myself, we have the stem cells in our skin, and that's why skin is continuously growing. In the bone marrow, we have hematopoietic stem cells. They are, because our blood continuously need supply and continuously need new cells, right? So these are the adult stem cells which appears in the 14 days old embryo and beyond, and we also have them, right? And they continue down certain lineages, but their problem is they cannot naturally turn back into the pluripotent or switch lineages. Switch lineages means here that if you are taking the skin stem cells, it will only give rise to skin. It will not give you the blood. If blood stem cells will only give rise to blood, it will not make the liver. However, the pluripotent and totipotent cell, they can give rise to any organ based on the microenvironment. Okay? So that is the most important concept. That multipotent stem cells, they are present in the adult, and they can give rise to only the cells of certain lineages, means certain types. However, pluripotent and totipotent cells, they can give rise, they have a lot of differentiation ability. They can give rise to multiple lineages, but then there's ethical issues, right? You cannot have the totipotent or pluripotent unless you damage the embryo, and you don't want to, ethically, it doesn't sound good to damage the embryo. However, multipotent stem cells easier to get, but they have certain limitations. So as I said, the blood, for example, the hematopoiesis. Hematopoiesis means that manufacturing of the different type of blood cells, and stem cells are multipotent. Here they can give rise to complete range of blood cell type, but if beyond the blood cells, they cannot make others. So commitment is a stepwise process. So here hematopoietic stem cells, here on the extreme right, you see all the cells, NK, T, B, dendritic cell, monocytes, neutrophils, they're all different type of blood cells. So one stem cell is giving rise to multiple different type of cell type, but only one catch. It can only give rise to blood related cells. It cannot give rise to the liver cells or heart cell or kidney cell. I am sure that last year or in 2019, you might have heard about this Chinese scientist who genetically edited babies. And there was a lot of controversy. He has his own story, but overall, entire world scientific community actually opposed and protest very strongly about him when he genetically edited 3 babies by saying that he is trying to make them resistant to HIV infection, while then people found out they were really not very prone to HIV anyway. So whatever he did, good or bad, all of all of us as scientists, we can condemn or somebody can favor, but this is still a topic to debate. For the person who is suffering, for them, it may be a very good thing that they can be treated and their life can be saved. But there are many issues in here which I think need more debate and more discussion. But although I completely understand to genetically modified babies to or making a genetically modified baby or the clones, but stem cell transplant has solved actually many problems. It's very fascinating that how so many kids, so many adults, their life are saved by the stem cell transplant who are suffering from many blood disorders. The easiest way to get the stem cell is the hematopoietic stem cells, because hematopoietic stem cells you can get from the bone marrow of any adult. And hematopoietic stem cells, they can treat any blood disorder because they can remake all the blood cell types, correct? So any disease related to the blood is treatable, whether it's a thalassemia, whether it's a sickle cell disease, whether this is a all different type of cancer originated from T cell, B cell, myeloid cells, whatever the blood cells cancers are, they are treatable cancer, and the treatment can be via the stem cell. So stem cell transplant work very well in the patients who are suffering from the blood disease. Here I will show you this experiment or briefly explain you how it works. Very similarly, it also works in the human being. So you have, let us say, this particular mouse. What you do is you irradiate this mouse, and irradiation basically removes the previous blood cells. Let us imagine a person is suffering from a blood disease, and here X-ray can irradiate or can deplete the old blood cell. And then you inject the bone marrow cells from the healthy donor. You take a donor, and if this is a sibling transplant, it is called autologous bone marrow transplant. Or if you take from a different individual, this is called allogenic transplant. I will not take you too many details that how much how different the individual should be. That is too many details. But imagine if it's the identical twins, it can be still come under there's the best allogenic transplant. Otherwise, you can take the bone marrow from the patients, but that will not be useful in all cases. In certain cases, it might be useful too. But allogenic means from a healthy donor, you take the healthy bone marrow cells and transfer into the patient. And then this mouse survives, and the entire blood will be changed now to the donor blood, because donor stem cells will now give rise to all blood cell type. Okay? So this is how stem cell transplant works in the normal individual, or in the cancer patient also, in the thalassemia patient also, who have the blood disorders. As a clinician, we identify a healthy donor for them, and then those healthy donors, we take the blood cells, take the bone marrow cells, inject into this person, and this person then ultimately its own cells goes away, and the stem cells from donor, it changed the blood. Now imagine this beautiful experiment, because this saved actually thousands and thousands of lives every day. Because in India, there's so many now bone marrow transplant centers, and this is done routinely in our country. So I hope you liked it, because this is very, very important experiment. And the final slide of my module is how you fix if you have certain genetic disorder. So imagine if a person is suffering from genetic disorder. Now you learned about the DNA. So you know that DNA is, if there's a change in the DNA, there could be a genetic disorder, and person can suffer many diseases. If the disorder is from one gene, one protein, then you can remove that particular gene and can engineer a new gene or correct gene copy, right? If there are 2 or 3 different genes are involved, you still can remove those defective copy of the gene, take the fresh gene, and this is called somatic gene therapy. Basically, you are trying to correct the defective copy of gene by replacing the correct gene using the lentiviruses or the retroviruses. And now here I want to now why I have taken this slide is I want to you to appreciate two concepts. One, DNA you learned, right? So you are basically, if there's a defective gene, Professor Sanjeeva told you about the cloning. So you remove that particular gene, add a fresh gene in that particular place in the chromosome. I have taught you the retroviruses, the RNA viruses. If you remember the life cycle of the retrovirus, when I was telling you, I told you that whenever the viruses come, they go and sit in your DNA. And when they sit in the DNA, they replicate. So this is exactly what, so using the virus as a vehicle. Here, we are using virus as a vehicle, and we are sending the correct copy of the gene using this virus as a vehicle. So virus is acting as a delivery vehicle, and our payload is the correct gene. And using the correct gene on the virus, we are sending into the human being, and these stem cells, we are sending on this stem cells, the correct copy of the gene will insert it into the correct place of the DNA in this patient, and the patient will be free of this disease. And here, what they are doing in this particular graph on the right, you are seeing that there is a hematopoietic stem cell. We are sending using the lentiviral vector, or you can just imagine it's a vehicle to send a correct copy of the gene. And these hematopoietic stem cells now will replace the defective blood cells or defective gene, and the person will feel good. So this is how your BB101 ends here, especially the lectures, and here there's tutorial lab